All right, everyone. So I'm in Seattle, and it's been fun. Hope you guys are having fun, not having class. Sorry about getting this out a little late. I am an idiot and forget that you guys are 3 hours ahead of me, so I clearly started recording this a little too late. But it's all right. You guys have the weekend to look over it too, so it shouldn't be too big of a deal. So today we're going to be talking about microbial genetics. Unfortunately, I don't have the ability to draw some pictures, so it might be a little bit difficult for some of the concepts, at least for me to explain them this way. However, I will include with this file a couple URL links to some YouTube videos that should help make some nice little pictures for you guys so it becomes a little more clear. Also, when I get back, we can do a quick little kind of synopsis of everything that I covered with this, especially some of the more, I don't know, conceptual stuff. So this is a somewhat long lecture. It's got about 80 slides. I'm going to skip over a couple bits and pieces here just so you guys aren't stuck listening to a movie that's an hour and a half long. But yeah, we'll see how this goes. Alright, so microbial genetics. So, okay, can't do that, sorry. Alright, so a little bit about microbial genetics as a whole. So basically it's the understanding of DNA, and as we've learned thus far, DNA is basically the sequence for life. All the DNA within a cell is considered the genome, and little bits and pieces of that genome are considered genes. Genes code for any number of things, usually proteins, enzymes, all that good stuff. And the compilation of genes that a organism has is what dictates its life, basically. So it's good to think of DNA as kind of like a brain, but it doesn't have direct control over itself. The DNA itself is what dictates what happens within the cell, and it doesn't actually control itself other than that. Bacterial genomes are fairly small. They have anywhere from 0.4 to 9.4 million nucleotides. So a nucleotide is just an individual segment, or not segment, an individual molecule of DNA. Those are the A, T, C's, and G's that I'm sure most people have heard of before. And then you can see in the human genome, and most eukaryotes, it's much, much larger, about 4 billion nucleotides. And the majority of these are just junk. 90% of our genome is junk and it does nothing. We don't know what it is. It gets spliced out eventually. So it's usually or generally considered to be just carryover material from evolution. So you can see that our genome is about 1,000 times larger than E. coli, but it only has 6 times more genes, and that's because the bacterial genomes are basically just all genes, no junk. A little bit of junk, but not that much. And there's no splicing out, so it's just whatever's there gets read, gets transcribed, translated, all that good stuff. So you can see for E. coli, 85% of their genome code for protein, where in our genome we get about 2%. So it's definitely not a bad thing to have a super or small genome, especially if you're a prokaryote. And then we'll talk about the differences between genotypic and phenotypic. So genotypic is basically the sequence of an organism's genome. So that's the A, T, C, and G content. Where the phenotypic is what you can physically see, or what the organism is physically manifesting. So if you can sit there and sequence an organism and look at all the different genes, and you can say that because they have this gene has this sequence, this animal will have a phenotype of red hair or something like that. So phenotypic is much easier, obviously, to comprehend and to also get a hold of, or to analyze, because it's usually something you can physically see. But genotypic tends to tell you a lot more information, almost too much information. It's getting to the point now where we can genotype organisms, or genotype even ourselves or other humans, but now we have so much information, like the whole entire genome or genetic code, and we don't really know what to do with it. So technology really is kind of at the forefront of genotype research. So the induction of genetics and genes. So genetics is the study of the inheritance or hereditary of living things. So most organisms that have sex, like us, basically transmit our genes to our offspring, but at different rates. And for that reason, there is a natural variety between all of us. Things like bacteria or smaller prokaryotes we'll be focusing on mainly in this lecture go through binary fusion or binary fission, and they don't actually mix up their genomic material between a mate. Instead, they just make exact copies of it and transmit it to their daughter cell that they create. One way that they get variety, though, is by mutations. So bacteria are a lot more easier to mutate, where we have a lot more mechanisms that prevent mutations. So we have to rely on our ability to have sex to increase variability between our genome and our neighbor's genome. So it's basically the transmission of traits from parents to offspring, expression and variation of these traits, so that would be the phenotypic variety or the phenotypic expression of these traits, the structure and function of the genetic material, and how this material changes. So it takes place in several levels, both the orgasmal, chromosomal, and molecular. So on the orgasmal level, you can see that's where the actual organism is composed of many cells, creating a tissue. The cellular level, you're getting variation between individual cells based on their genetic material. On the chromosomal level, you're getting the variation between the different pieces of DNA. And then on the molecular level, this stupid thing. Okay, on the molecular level, you're getting variation between the A, T, C's, and G content. So the nature of genetic material must be able to self-replicate. So if the genetic material can't actually replicate itself, the cell can't replicate. You need to be able to make a duplicate copy of your DNA so that when you create a new cell, it has the same genetic code as you did. And it must be accurately duplicated and separated from each daughter cell. However, things like prokaryotes and viruses tend to mutate much more readily than eukaryotes, especially viruses. Viruses are notorious for mutations, and that's one of their ways of evolving. That's how they tend to become super pathogens, such as HIV. So level and structure of the genome. So the genome is basically all the genetic material within a cell. Then you have bits and pieces of that genome where it's broken down into segments, and those are called chromosomes. And these compose of anywhere from thousands to hundreds of thousands of genes on each chromosome. And then to break it down a little further, you have genes, and genes usually code for either one or a handful of different proteins and enzymes. You can have things that code for structural things like actin, microtubules, all that good stuff. Genes for RNA machinery, so this is where it gets transcribed directly into things, or right into our RNA, which then can be made into ribosomes and such for making proteins. And then you also have regulatory genes, so you can have one gene that once it's turned on, it turns on a series of genes after it. And usually these genes are all part of a gene family, which code for something physiologically important for each of those genes, so they're all related somehow. So size and packaging of genome, so they vary greatly in size. Smallest viruses have about 4 to 5 genes. Again, you have to remember that viruses need to hijack a cellular's machinery, or the cell that it's infecting machinery. So it doesn't really need that many genes. It just needs enough genes to A, get it inside of the cell, and B, allow it to take over. So 4 or 5 are plenty. E. coli has about 4,288. That's because it basically lives a life where it doesn't require another organism. It doesn't need to take over cellular machinery, so it needs to have enough genes to code every protein that it's going to require to survive. And then human cells have about 20,000 to 25,000 genes, most of which don't really do anything, or we don't know what they do yet, but they are there. So the stretched out DNA can be 1,000 times more longer than the cell itself. So for that reason, we have to compact the DNA, and that's usually by wrapping them around histones. So in eukaryotic cells, going into chromosomes, so a chromosome is essentially condensed down DNA. So if you were to just let the DNA be loose and open, it would just take up the entire portion of the interior of the cell. So for that, we need to pack it down, and that's usually done by wrapping the DNA around histones in multiple different levels. So you have your helical DNA, which then gets wrapped around a histone 3 protein. Then those histone 3 proteins then wrap around another larger histone protein. And once that happens, you get this nice condensed down chromosome. Those you can physically see with your eyes under a light microscope, so they are fairly large. They're always located within the nucleus, and they vary in number from a few to hundreds. We have 46, but there are some yeast out there that have, like, I think one species of yeast has like 2,000 chromosomes. So just because you have more chromosomes doesn't mean you're a higher order organism. It just means you've evolved and mutated slightly differently than another organism with smaller amount of chromosomes. They can occur in pairs like us, or haploid singles. So if it occurs in pairs, that means the organism reproduces via sex. So it has one set of chromosomes from mom, one set of chromosomes from dad. And each set of chromosomes has the same genes on there. The genes are just slightly different from mom and slightly different from dad, and whichever one of those genes gets turned on, usually by happenstance, that is what gene gets produced, and that's what it produces the person. And that's why people are unique. They're not the same as their parents, because it's a mixture of both genetic materials. Haploid organisms, on the other hand, are either gametes, or the actual reproductive cells of sexual organisms like ourselves, or they're asexual organisms like a lot of fungi and lower eukaryotes. And they appear to be linear, just broken up linear segments. Our chromosomes are in a nice little X shape held together by a centromere, but some higher order eukaryotes doesn't necessarily look like an X. Just depends on the organism. Bacterial chromosomes, on the other hand, they're condensed and secured by means of histone-like proteins, so they're not exactly histones, but they serve a similar function, so we give them the name histone-like. And it's a single circular chromosome, which is pretty easy to do if you have a small genome like a bacteria. So gene is a certain segment of DNA that contains the necessary code to make a protein or RNA molecule. So this is the bit that actually gets transcribed, translated into a protein. They're regulatory genes, so these control gene expression, and usually they're in front of a series of genes that they control. And sum of all types in an organism is called the genotype. So if you take all of the genes that are either turned on or turned off throughout a cell's life, that would compile the genotype. The expression of these genotypes create traits, and these are the phenotypes. These are the things that we can physically see, whether that is, someone's hair color, or going into a cell and looking at what proteins are created using molecular biological techniques. Both of them are considered phenotypes because you're looking at something after it has been transcribed and translated. And then all organisms contain more genes in their genotype than are manifested as a phenotype at any given time. And that's because our cells have the ability of saying, okay, we need these genes turned on at this point, but if conditions change or something drastically happens to that cell, then maybe another series of genes will be turned on. And this is all done by regulation, which we'll explain in a little bit. So the DNA code is just a simple yet profound message. So it was discovered in 1953 by Watson and Crick. It's basically made up of 4 nucleic acids and 2 strands combined into a double helical form. So it's this combination of all these, or these 4 different nucleic acids in different sequences, and this gives the wide variety of life we see today. So the general structure of DNA, the basic unit is called a nucleotide. So this is the A, T, C, and G by themselves. The basic backbone of a nucleotide is the phosphate, followed by deoxyribose sugar, and then the nitrogenous base. The nitrogenous base is what varies. That's what makes your A, T, C, and G. These are covalently bonded together to form a sugar-phosphate linkage, the backbone of each strand. One bond is to the 5' carbon on the deoxyribose, the other is to the 3' carbon. So this allows you to create a single or a strand of nucleotides. And these strands of nucleotides are what compose of genes. And because they have this nice pi covalent bond between them, it becomes very hard to pull apart. And this is what prevents mutations from occurring. Because if you have a nice sequence and the sequence codes for a gene that you really, really need, if that sequence can be broken up by these bonds breaking, you then would have some sort of mutation because you wouldn't get that gene being produced the way that it should be. That mutation would obviously be bad because that gene is required for life. So it's important to keep these nice covalent bonds between these nucleotides so that the sequence doesn't get mushed up or switched. But it does happen, and it's usually because of forces that are strong enough to break these covalent bonds, such as UV radiation or heat or some elements like oxygen or oxidizers. So here's a kind of a molecular picture of DNA. You can see the A, C's, G's, and T's. This is what composes the nitrogenous bases, followed by the deoxyribose and then the phosphate bond. Here you can see these nice covalent bonds that bind the nucleotides together into a sequence, and then you have one strand here, and then you have another strand of the DNA here, and they're held together by hydrogen bonds. Now, hydrogen bonds are weak. They're the weakest bond around, basically. And for that reason, they're pretty easy to pull apart. If you pull these hydrogen bonds apart, you can then make a copy of either strand of DNA. And the cell utilizes this to create identical copies of its genome when it's duplicating. You can see here between G's and C's, you have 3 hydrogen bonds, and between T's and A's, you have 2 hydrogen bonds. So it's weaker at the T and A bond site. So whenever you want to start duplicating your DNA and you need to kind of separate these 2 strands, it tends to happen at a TA rich site. And that's because these are weaker and it's easier for helicase to get in and break them apart. So nitrogenous bases, you have your purines and your pyrimidines. They span the center of the molecule and pair with complementary bases. These pairs are called base pairs. Paired bases are joined by hydrogen bonds, which are easily broken and allow the molecule to be somewhat unzipped, allowing for DNA replication or transcription. Adenine and guanine are your purines. Thymidine and cytosine are your pyrimidines. And adenine is always paired with thymidine, and guanine is always paired with cytosine. So you have A to T and G to C, always. Oops. Anti-parallel arrangement. So you basically have 2 strands of DNA, one running in the 5' to 3' direction, which is basically like reading from left to right, like we do now. And then you have another strand that's complementary to that, running in the opposite direction. So it's running from 3' to 5'. So it's running in a backwards direction. This just allows for Alright, I'm back. So my computer froze in the middle of the last recording, so it will be in 2 recordings, unfortunately. Well, hopefully it doesn't freeze again, or it will be in 3. But we'll just go right where I left off from before. So the last slide that we talked about was this one. So now we're going to go into DNA replication and passing on genetic material. So duplication is exactly what it sounds like. It's making an identical copy of the genome so that it can be passed on to the new daughter cell or offspring. So it must be completed during a single generation time, so it needs to be fairly quick. And for that reason, there are many enzymes working at any given time doing the replication process. Some cells, DNA replication can happen in a matter of minutes. I know for T cells it only takes about a couple hours, which is pretty nice considering they're a human cell and they have a fairly large genome. Bacterial cells, E. coli can do it in less than a minute. So it's fairly quick, fairly rapid for smaller genomes. This requires the action of 30 different enzymes, which I'm not going to expect you to know all of. There's just a handful that are important for what we're talking about. And the basic concept is to separate the strands. So you have double-stranded DNA. You need to separate them into the two compartments, or the two components, the 5' to 3' sense strand and the 3' to 5' antisense strand. You then copy both of those templates by adding new free nucleotides to both single-stranded pieces, making two single-stranded copies of, or two double-stranded copies of the DNA. And then this will produce two daughter molecules, which then can go, one will go to the daughter cell, one will stay with the parental cell. So a couple of the important enzymes I'll expect you to know. Helicase, so this is for unzipping of the DNA helix. This binds to the A and T rich sites, breaks those double bonds, and then kind of zips along the double-stranded DNA, breaking it up into its two separate pieces. Primase, so this adds an RNA primer. This RNA primer is what tells DNA polymerase where to bind. Once DNA polymerase binds, it can then grab free nucleotides that are in the cytosol and add them to the single-stranded piece of DNA. DNA polymerase 3 is the one that adds the bases to the new DNA chain. DNA polymerase 1 removes the primer, so this removes the RNA primer that primase placed, and then it closes the gaps by adding nucleotides in that bit where the primer was bound. Ligase then comes through and closes all of the gaps between the deoxyribose sugars in the sequence. So when DNA polymerase 1 comes through and fills in the gap where the RNA primer was, it just puts in nucleotides, but it doesn't actually make the covalent bonds between the deoxyriboses. So ligase will come through and kind of push them together, creating these nice covalent bonds. And topoimerase 1 and 2, so this is for supercoiling and untangling. So these are kind of larger proteins that will kind of prevent the DNA molecule from kind of folding in on itself, allowing it to be open. And by being open, it allows for DNA polymerase to bind and add in nucleotides. So the most simplest form of replication, which happens with small genomes, so you see this a lot in prokaryotes, is semi-conservative replication. So each strand acts as a template. So here's our double-stranded DNA. We break apart the weak hydrogen bonds with helicase, creating two single-stranded pieces of DNA. Then you add in DNA polymerase 3, which binds to the RNA primer that was laid by primase, and it reads in a 5' to 3' direction on both sides. So it kind of works in opposite directions. This works really well if you have a circular genome, because you can kind of start at one end, start at the other end, and then just kind of go zip around until you reach the other end of the circular genome. If you don't have a circular genome like in us, this becomes almost impossible and kind of a waste of energy. So we do something a little bit different and a lot more complex. This allows for accurate replication, but again, it is somewhat slow, especially if you have a large genome. If you have a small genome, it's perfectly fine. The biggest thing to take from this slide is that DNA polymerase 3 reads only in the 5' to 3' direction. It will never go in a 3' to 5' direction. So for that reason, it has to go this direction, and then it has to go this direction on the opposite strands. So it's going from 5' to 3' this way, and then 5' to 3' this way. So the starting bit is the origin of replication. So that's the site where helicase will bind and start the unzipping. So it's a short sequence rich in A's and T's. These A's and T's only have 2 hydrogen bonds, allowing for a more weak interaction. So where helicase can kind of just plow right through. It's held together by only 2 H bonds rather than 3, obviously, and then less energy is required. The helicase binds to the DNA at that origin, untwists the helix, breaks the hydrogen bonds, and results in 2 separate strands of DNA. Here's another video. You can check that out at your leisure. I won't play it because it just wastes memory. So DNA polymerase synthesizes the new daughter strand by basically grabbing free nucleotides, and these nucleotides will bind to their counterpart. So A to T, C to G, and vice versa. The process depends on several other enzymes as well, but key points about DNA polymerase 3. So it does require a whole bunch of different factors that kind of bind to DNA polymerase and allow for this process to happen. But for our purposes, I just expect you to know what DNA polymerase 3 is. So nucleotides that need to be read by DNA polymerase are buried in the double helix. So the DNA must first be unwound and 2 strands separated. So that makes sense. If they're in their double-stranded positioning, those nitrogenous bases are hidden, and it makes it impossible for free nucleotides to bind. DNA polymerase 3 is then unable to begin synthesizing. That's where helicase comes in and breaks these hydrogen bonds, allowing for DNA polymerase to bind. And it can only add from a 5' to 3' direction. And these nucleotides are regularly synthesized by the cell, or they're brought in from the environment. So there's always free nucleotides kind of floating around for DNA polymerase 3 to grab a hold of and create these new strands. So as the replication proceeds, new double strand begins. DNA polymerase 1 then removes the RNA primer that primase laid down, and then replaces it with DNA. Once this happens, like I said, you have these little gaps between the deoxyriboses, so the covalent bonds aren't fully formed yet, and that's where ligase comes in and fuses these together, creating a nice, solid, strong piece of DNA. Oh, now it's going. My computer keeps acting up and freezing, so I don't know what the deal is. Now, this is a little bit more complex. So before we were talking about, basically you have one circular chromosome, like here. And you put one DNA polymerase 3 here, one DNA polymerase here, and they go in the 5' to 3' direction in opposite directions until you get 2 single-stranded pieces, or 2 double-stranded chromosomes that are copies of each other. If you want to speed up the process, you can put 4 DNA polymerases, so 2 on each single strand, and then they will move in opposite directions. But the problem with this is that you can only read in a 5' to 3' direction. So if my 5' to 3' direction is here, that means this guy can read perfectly fine, but this guy can't, because he can only go in this direction. So he can start moving in this direction, but he'll run into the double-stranded DNA that this DNA polymerase created instead of going in this direction. So a way around this is by using the enzyme primase, and the primase will then travel down this strand and place RNA primers in random locations, about 100 nucleotides or so away from each other. And what this does is say, okay, DNA polymerase 3, every time you see an RNA primer, start adding nucleotides. So it can then go down the line in the opposite direction, find an RNA primer, bind, then fill in the bits in between, going in the 5', 3' direction. Unbind, go back, find the next RNA, which you can see here. So here you have this guy, he's going in the 5', 3' direction without a problem. But this guy, he can't do that, obviously. Going in this direction is 3' to 5'. It's impossible for DNA polymerase to do that. So these little triangles here represent the RNA primers that were added by RNA primase. And this basically says, okay, DNA polymerase, bind at these RNA primers and then fill in until you reach double-stranded DNA. go to the next one, fill in until you reach double-stranded DNA, and go for on and on and on and on. That's when DNA polymerase 1 comes in, removes these RNA primers, and adds in nucleotides, but there's still little gaps in between the deoxyriboses, and that's when ligase comes in, fuses these deoxyribose gaps, allowing for a strong segment of double-stranded DNA. Here you can see this is what happens as the DNA is being copied. You get these two identical copies of the circular chromosome, and at the very end you get something called a nick, and this little nick eventually breaks off, allowing for two separate copies of DNA. Here's another movie you can watch at your leisure. Don't worry about that. Alright, so applications of the DNA code, so transcription and translation. So now we've made copies of our DNA, meaning we can make copies of ourselves. But the important, or the most important, part of the cell life cycle is producing proteins. If you don't produce proteins, you don't survive. So the central dogma behind all of this is taking DNA, making an mRNA copy of a gene, and then taking that mRNA copy and translating it into a protein. So to go from DNA to RNA is called transcription. You've heard me talk about this before, so it should be just review. Then going from mRNA to actual protein is translation. Transcription is fairly easy to understand, and that will be a real easy, quick talk point for me. Translation is a little bit more complex, and we'll probably have to go over that when I get back on the board, just because it makes it a little easier to see or to understand when you can see it. Some exceptions to this obviously are RNA viruses. So they have their genome is actually RNA instead of DNA. So they obviously don't have to go from DNA to RNA since they're already RNA. Okay. In fact, some retroviruses will go from RNA to DNA and then from DNA to RNA. So they're a little different. So in addition to RNA that produces proteins, other RNAs are used to regulate gene function. These are called shRNAs. They're just small RNA fragments that actually kind of interfere with translation, and they're used as a way for the cell to control how much protein is being produced. We also utilize them in the lab to shut proteins down and then see what happens to the cell after that protein is gone. And then many of the genetic malfunctions that cause human disease are found in these regulatory RNA segments. So here's just kind of a picture of the central dogma. You have your DNA, you transcribe it into mRNA or other versions of RNA. tRNA is what aids in translation. rRNA is what makes up the ribosome. But most of the time it's transcribed into mRNA. mRNA in a eukaryote will then translocate outside of the nucleus, where it can then be translated via a ribosome. If you're in a prokaryotic cell, this all happens almost simultaneously. You'll get your mRNA transcript, then you'll translate it right in the same spot as the DNA, because there's no nucleus, obviously. During translation, you're adding on amino acids until you form a nice amino acid strand called a peptide. And these peptides get longer and longer and longer, and then they kind of get folded by different cellular machinery like the Golgi and endoplasmic reticulum until you get your final product of a protein. So gene-protein connection, so the triplet code and the relationship to proteins. So 3 consecutive bases of DNA strands are called triplets. These triplets code for an amino acid. There is redundancy in the code, so multiple triplet codes, or codons as they're called, can code for one amino acid. So this allows for mutations. This prevents mutations from having a super great effect all the time. That's because if you just do a point mutation where you just change one amino acid, the chances of you changing it so that it becomes a totally different amino acid are reduced by 3, essentially. It's transcribed and translated, and it dictates the type and order of amino acids in a polypeptide chain. Proteins ultimately determine phenotype, and DNA is mainly a blueprint. Like we've mentioned before, it's the basis of, it's the code, it's what describes everything. So here we have DNA, it's being transcribed into mRNA, then we have amino acids during translation that each one of these amino acids code for 3 nucleotides, or 3 nucleotides code for 1 amino acid. And these 3 nucleotide segments are called codons, and there's some redundancy built into these. So RNA is essentially almost the same thing as DNA, except instead of a deoxyribose sugar backbone, it has a ribose sugar backbone. Can be in helical form, can be in single-stranded form. It doesn't have T or thymine, it has uracils. So instead of AC, T, and G, it has AC, U, and G. And the sugar is ribose, obviously. A transcript of a structural gene, that's what mRNA is. It's basically a RNA copy of the DNA. It's as simple as that. The reason we do this is because A, we want control over what's going to be produced. Having an mRNA message is one way of controlling this, especially in eukaryotic cells. Just because we produce an mRNA message doesn't mean it's going to get translated. It first has to be translocated out of the nucleus before it becomes translated, so it's a way of controlling. Also, mRNA isn't forever. It is degraded. So if it gets degraded at a fairly regular rate, then we don't have to worry about too much of that protein being expressed. Also, your DNA is very special. You don't want it to be constantly played with or manipulated, and that is what would happen if we used it to directly translate to protein. You want a filler message in between there that isn't that special. If mRNA gets degraded, not a big deal, we'll just make more. If the genome got degraded, there's nowhere to go. You can't make more. It's a problem. So transfer RNA or tRNA, so this is a 75 to 95 nucleotide long length piece of RNA, and this RNA has something called an anticodon built into it, and the anticodon is what binds to the codon of the mRNA. And so this anticodon will bind to a specific codon, and attached to the other side of the tRNA is an amino acid. So this is what allows for the tRNA to bind to the mRNA, putting the amino acids close to each other, allowing them to bind together, forming a peptide or a piece of a protein. So it's where the T comes from. It actually transfers amino acids from the cytosol to a ribosome and allow them to bind to their corresponding codon sequence on an mRNA segment. And there are 20 amino acids, and 20 amino acids, different combinations of those are what create different proteins. So here are the different RNA molecules. mRNA, you can see it just looks like a single-stranded piece of, or single-stranded molecule of nucleic acids. And then transfer RNA has got this kind of secondary structure. It looks like a T. You can see the anticodon here. This is what binds the codon of the mRNA. And then here you have the amino acid attached to it. So during translation, this guy will come up, bind to its corresponding codon, and its amino acid will then bind to a neighboring amino acid that is being bound to this codon. And by this happening over and over and over and over and over again, you get a nice linear amino acid polypeptide. So the ribosome, it's made up of rRNA. It's essentially got 3 different sites, the EPA sites. It's pretty easy to remember, Environmental Protection Agency. You need something to help you remember it. Prokaryotic 70S ribosome, so it's composed of tightly packed rRNA. It's a little smaller than the eukaryotic ribosome, and it's definitely different, and this is a good target for a lot of antibiotics. So the RNA component is a long polynucleotide molecule, but it's very, very large. But again, it has to be made out of RNA, because if the ribosome was made out of protein, and the ribosome is the only thing that can make protein, it obviously can't make itself, so you run into problems. And the interactions of the protein and RNA create the 2 subunits of the ribosome. that engages in the final translation of genetic code. So you have two subunits, the large subunit and the small subunit. The mRNA segment right here will kind of bind in between. And then you have your tRNAs binding with their anticodons, binding to the codons. And this puts two amino acids in close proximity. These amino acids can then bind, creating a polypeptide. And then what ends up happening is this guy gets ratcheted down so that this site will then move down here, this site will then move down here, and then you'll have this site here in this new A site, allowing for a new tRNA to bind. So it's basically like a chain reaction that occurs. So transcription is fairly easy. You have RNA polymerase II. It binds to the double-stranded DNA, it unzips the double-stranded DNA, and it adds free-floating RNA nucleotides, creating an mRNA transcript. Now there are other enzymes that do aid in this, but for our purposes, I just expect you to know RNA polymerase II. And he kind of does everything. He binds, he unzips, he adds in free new RNA nucleotides, creating the mRNA transcript. It's as simple as that. Here's a movie. Translation. Now this is the second stage of gene expression. This is where you're creating a protein from the mRNA transcript. So all the elements need to be synthesized. Proteins are brought together by the ribosome. There's 5 stages: initiation, elongation, termination, protein folding, and then protein processing. Alright guys, so I'm going to try this one more time. So this is the third part of the lecture. My program crashed spontaneously again for like the 6th time, so it's driving me nuts. But hopefully this will be the last time, but we will start off where we left off from before. So now we'll talk about transcription. It's basically broken up into 5 different stages: initiation, elongation, termination, protein folding, and protein processing. Initiation is dictated by the start site on the mRNA transcript. This says, hey, ribosome, come bind here. It tells where the ribosome to go. Elongation is where you actually create your polypeptide, or your single strand of amino acids. And this is called elongation because you're elongating your strand of amino acids. Termination is dictated by a stop codon, and this says, okay, ribosome, we're done making this polypeptide, it's finished, unbind and release the polypeptide. That single linear piece of polypeptide can then be folded by enzymes in prokaryotes in the cytosol and eukaryotes in the Golgi, and this is where the protein actually takes its shape. Its 3D shape, I should say. And then protein processing are post-translational modifications. So these allow the protein to be modified after translation to give it a better or more specific function. So initiation of translation. So mRNA molecule leaves the DNA transcription site. It's then transported to the ribosome in the cytoplasm. Ribosome subunits are specifically adapted to assembling and forming sites to hold mRNA transcript at the start codon. In eukaryotes, the mRNA transcript must translocate or leave the nucleus and go out into the cytoplasm. In prokaryotes, it happens simultaneously. So right as transcription's occurring, translation's occurring, and proteins being produced. So the messenger RNA is the master, or the message, or from the master genetic code, or from the genome, the DNA, and it's full of codons, and these codons have redundancies. So you can see here, here's one amino acid, leucine. It has 4 different codons that could code for leucine. You can see CUU, CUA. So if there's one small mutation within this codon, changing this U to an A, you would still get leucine and nothing would happen. You'd be fine. So for that reason, these redundancies make point mutations a lot less severe. So it's read in triplets, or a codon. So you can see here we have the DNA. It's transcribed into RNA. There are RNA codons. And then there are anticodons on the tRNA that bind to these codons, and carrying along with the tRNA as an amino acid. They come into close proximity with each other, allowing for covalent bonds to be formed between them, creating the polypeptide chain. There's 64 possibilities because there are four possible codons for each amino acid. Oops. And it's going backwards. So, translation. This is it in a nutshell. Basically, you have initiation, which starts with the mRNA being bound by the two subunits of the ribosome, the large subunit and the small subunit. This happens at the start site, which is right here. You then have two codons of that mRNA sequence being exposed to the P and the A site in the ribosome. This allows for tRNA with its associated amino acid attached to bind to these codon sequences. Once they bind, you get a peptide bond between the two amino acids because they're in close proximity to each other. And occasionally there's enzymes around that kind of help this bond form. Once this bond forms, you get one amino acid jumping from this tRNA to the other amino acid on this tRNA. So this tRNA is now empty, no longer has an amino acid, and it leaves via the E site. So it goes from the P site to the E site. This guy on the A site then moves over to the P site. And while he's moving over, he takes the mRNA with it. This forces the mRNA up one codon. So you can see here, this guy in the A site, right here, travels over to the P site, bringing with it the mRNA, ratcheting it up one codon. This exposes a new codon in the A site, allowing for tRNA and its corresponding anticodon and amino acid to bind to this codon, allowing for a new amino acid to come into close proximity to this new chain of amino acids. A new peptide bond is formed, creating 3 amino acids, a 3 amino acid chain. You then have that guy in the 3 amino acid chain in the A site moving down to the P site and bringing along with it the mRNA strand, ratcheting it one codon forward, exposing a new codon. And this process happens over and over and over and over again until you get to a stop codon. Once you get to the stop codon, the ribosome says, okay, this polypeptide is done. Let's go the mRNA, and releases the polypeptide so that it can be folded. Here's the video if you want to watch the video. Termination, so it's brought on by UAA, UAG, or UGA. These are all stop codons. If they're called nonsense codons, there's also something called a nonsense mutation, and that's where you mutate a codon so that it becomes a stop codon, essentially ending translation, stopping the protein from being produced, and those are very, very severe. Some of them actually cause pretty serious diseases. And then when reached, a special enzyme breaks the bonds between the final tRNA and the finished polypeptide chain, releasing it from the ribosome. So modifications to proteins, you can have post-translational modifications. You can clip off amino acids, you can add cofactors, you can add in different groups, different molecules that are found within the environment. And you can also join proteins together, making a large protein with many units. So in prokaryotes, this all happens simultaneously. So you have your RNA polymerase making the mRNA transcript, and before that mRNA transcript can actually fall off from the DNA, you have ribosomes binding and producing protein. This is why it's very easy for bacteria to produce protein, and they produce it fairly quickly. So it all happens simultaneously. In eukaryotes, start codon is an AUG site, but it codes for a different form of methylene. Don't worry about that. It's just a different site. That's all I expect you to know. And mRNA transcripts code for only one protein. In prokaryotes, you have a very long mRNA transcript that codes for many proteins. In eukaryotes, it's just one protein that each mRNA codes for. Since DNA is in the nucleus, the mRNA must leave the nucleus, so it translocates out into the cytoplasm. And as it's going through the cytoplasm, it gets spliced. So I mentioned earlier that a lot of the DNA in our genome, or a lot of the sequences in our genome, are nonsensical, so they make no sense. They don't code for anything. So those need to be taken out of the mRNA transcript. And that happens as it's leaving the nucleus. So eukaryotic split genes, so introns, so these are bits that do not code for anything. So this is the nonsensical stuff. This is the majority of our genome, useless information. Exons are what code for proteins, but they're not in a nice segregated fashion. So you don't have just one long exon that codes for a gene and then a whole bunch of introns that don't code for anything. Instead, you have a little bit of an exon here, a little bit of an intron, a little bit of exon, a little bit of an intron, a little bit of an exon, and so forth. And you actually have to splice these guys out, like down here, and fuse the exons together to actually get the mRNA transcript. And that's what occurs in the nucleus right before it leaves the cytoplasm. Here's a video. You can watch that. Another video. Watch that. Now on to viruses. So viruses are very diverse. So some nucleic acid is linear, others circular, just depends on the virus. Most exist in a single molecule. It's not broken up into segments. However, there are a few, such as influenza, that are broken up into segments. Most contain double-stranded DNA or single-stranded RNA, but there are other patterns. And as we saw before when we talked about viruses, most have single-stranded RNA. It's the most common. In all cases, viral nucleic acid penetrates the cell. Obviously, it needs to get into a cell so that it can hijack the cellular machinery and parasitize the cell itself. Nucleic acid is introduced in the host gene processing machinery. The virus instructs the host machinery to synthesize large numbers of new virus particles and not focus on the actual cellular proteins. And then viral mRNA is translated into protein. The mRNA is then taken to the ribosome, uses tRNA and amino acids from the host, creating viral proteins. These viral proteins then kind of self-congregate, creating new molecules or viruses, and those bud off or explode out of the cell. How do we regulate genes? So there are many, many different ways. We'll talk about the most classical example here. So there's two different types, inducible or repressible. So an inducible is where you have an operon or a small portion of the gene that actually dictates if the gene is going to be turned on or turned off. And you have an operon that is turned on when substrate binds. So if you have substrate present, it will turn on the gene. Repressible, on the other hand, is if substrate's present that binds to the operon, it turns off the gene. So this works in a negative feedback loop. Inducible works in a positive feedback loop. So an example of a positive feedback loop is for lactose, or the lactose operon in E. coli. So the lactose operon is within the lactose locus. So a locus is basically a location on the chromosome for a specific gene. It's kind of like the roadmap to where the gene is in the genome. In the gene itself, you have a promoter, so this is recognized by RNA polymerase. This is what allows for transcription to occur. You also have an operator. This is a sequence that turns on or turns off the gene. So for lactose, there are 3 genes that need to be turned on so that E. coli can metabolize lactose properly. So here you can see the promoter, the operator, and then the 3 structural genes. If lactose is not present, you have this repressor protein that binds to the operator, preventing RNA polymerase from transcribing. But if, for whatever reason, E. coli comes into contact with lactose and it now needs to metabolize lactose, lactose will come into the cytosol, it will bind to this repressor protein. The repressor protein will then disassociate or leave the operator, allowing for RNA polymerase to fully transcribe the 3 structural genes into mRNA. These mRNA, or this mRNA, can then be translated into proteins. These proteins happen to code for enzymes that will digest or break down lactose. Once it breaks down all of the lactose in the cytoplasm, and no more lactose is coming into the cell, this repressor can then bind back to the operator, preventing the lactose or lactase enzymes from being produced. Here's a movie of that if you're interested. A repressor operon, on the other hand, works in a negative feedback loop. So that's where whatever is being produced will bind to the repressor and actually turn the repressor on, stopping transcription from occurring. So here you can see in a normal situation, you have RNA polymerase binding to the promoter and transcribing these 3 genes into an mRNA. The mRNA can be translated into protein. Now the product of this protein happens to be some, I don't know, in our case it looks like a little diamond-shaped protein, I don't know, whatever that is. Now when these diamond-shaped proteins build up to a critical level, they will bind to a repressor molecule. Once this repressor molecule has this cofactor bound to it, or the actual substrate we were producing from our enzymes before, it becomes activated and then it binds to this operator, preventing transcription. So this allows for enzymes being produced, but once a certain number of those enzymes are produced and they're producing enough or modifying enough substrate to where we don't need them anymore, that substrate can then bind to a repressor protein, activate it, and stop those enzymes from being produced. So it's a way of controlling the amount of enzyme present. Antibiotics that affect transcription and translation. So this is a very common way for antibiotics to work in prokaryotic cells. So we have certain drugs that interact with DNA, RNA, ribosomes, and again, ribosomes are a good target because they are slightly different than our ribosomes. This reduces the amount of cytotoxicity that occurs with a lot of antibiotics, making them more effective and less toxic to us. Don't worry about that. Alright, so what are mutations? So mutation is a switch up in the genetic code. This is usually because the covalent bonds between the individual nucleic acids or nucleotides that form the sequence somehow get broken or get mismatched. So mutation is a change in nucleotide sequence, where wild type is a microorganism that exhibits the natural non-mutated characteristic. A mutant strain is the microorganism that has the mutation. So generally when you say wild type, you're talking about a normal animal, human, whatever the case may be. A mutant strain is where you've mutated something on purpose, at least in a laboratory setting. So there are a lot of mutation tests. They use these to see if chemicals are mutagens, or things that cause mutations in animals or prokaryotes. And the easiest way to do this is with something called an Ames test. And you basically take bacteria and put them in the mutation bit and allow the mutations to occur. If the mutations occur in these specialized bacteria that are lacking some key enzymes, you have a higher chance of spontaneously turning on these key enzymes. So if these key enzymes are turned on, the bacteria will reproduce. If these key enzymes are not turned on, the bacteria will not reproduce. So since mutations occur at a fairly regular rate, especially in bacteria because they mutate often, especially if they're exposed to a really good mutagen, you'll get mutations, so many of them that these enzymes turn back on and they can reproduce. So in a nutshell, what happens is if mutations are occurring, you get a lot of bacteria. If mutations aren't occurring, you get no bacteria. So spontaneous mutation is just a random change in DNA arising from errors in replication. So this can be due to enzymes, the replication enzymes. It can be due to chemicals, and it can be due to things like UV radiation or gamma radiation, like radioactive waste and material and all that. And then induced mutations are results exposures to known mutagens. So what kind of mutations are there? There are point mutations. So point mutations involve addition, deletion, or substitution of a single base. These are usually much less severe, and that's because we do have the redundancy in codons. So you just change one nucleotide or amino acid. It doesn't really, or one nucleotide in the DNA itself, there's a good chance that it's not going to really change the amino acid that corresponds with that codon. So missense mutation is any change in the code that leads to a placement of a different amino acid. These happen somewhat regularly, especially in lower ordered organisms like prokaryotes and viruses. Nonsense mutations is where you change from a normal codon to a stop codon. So out of all the point mutations, this is the most severe because it basically stops the translation of that mRNA sequence before it can actually elongate to its full form. Silent mutations occur, so this is where base change, but it does not change the amino acid. So it doesn't actually, it changes the codon, but the codon still codes for the same amino acid. And then back mutations are when a gene that has undergone mutation reverses to its original base composition. And this does happen fairly regularly in lower organisms that mutate on a regular basis. Now frame shift mutations are much, much more severe, and this is where you actually delete or add usually a couple nucleotides, and by doing this you change the entire reading frame. So if I have a gene that's 100 base pairs in length and it needs to be read from 1 to 100 to make the actual protein, if I delete maybe 3 amino acids in the middle, I now have a gene that is 1 to 97 nucleotides in length. And everything that happened after that deletion is now 3 nucleotides ahead. So it's not going to be coded in the right way. So the codons are going to be out of order. And this basically obliterates the protein. The protein doesn't work. You don't get the right amino acid sequence. So here's normal gene up here, missense mutations. So this is leading to amino acid switch. Now it does change the amino acid, but it's not always necessarily a bad thing. Just depends on how severe or where it's located in the protein. Base substitutions or silent mutations, so no change happens. And from here we go from an A to a U. Since there's redundancy in the codons, we still stay with aspartum. Here we have a frame shift mutation where we're deleting something, so we'll delete this C and it then gets filled with a G, and this changes everything after it. You basically got rid of one codon and moved the stop codon earlier, so it kind of just screws up the entire protein after that mutation. So repairing of mutations. So most ordinary DNA damage is resolved by enzymatic systems, especially in eukaryotic cells. We have fairly good enzymatic reactions that can actually protect and repair our DNA, especially from things like UV radiation, because we're in constant bombardment by UV light. So DNA photolase is a light-sensitive enzyme that gets turned on when UV light is present, and this kind of turns in the DNA repair, or turns on the DNA repair mechanisms. And then excision repair is actually where you excise a portion of DNA that is just too badly damaged to repair and replace it with new. Another picture. Here's the Ames test. So this is what I alluded to earlier, but this is a more detailed description. So you basically have bacteria, this salmonella bacteria that is missing its histidine enzyme. So what you're basically saying is if there are no mutations occurring in this organism, you will get no growth because they don't have histidine and they don't have an enzyme that processes histidine, and that is absolutely required for the organism to survive. Now, if you do add something into these bacteria and it causes a lot of mutations, because bacteria mutate at a fairly regular rate or fairly quickly, and there are a lot of them, you can expect that there are going to be some mutations that occur in the enzyme that is screwed up by us, the histidine enzyme, and it will actually correct the enzyme and make it work. And once it corrects the enzyme so that it works, it allows for growth. So the more colonies you get, the more mutations that are occurring. So it's a little counterintuitive, but But it's a way for us to say, okay, we have this drug or we have this chemical and we want to know if it's going to cause cancer down the line because it's a mutagen. And the one way to test that is by doing this test. Granted, it's in bacteria and not in eukaryotes or in our cells or even an animal or anything, but it is kind of an easy, quick method to get an idea if something is going to be a mutagen or not. So positive and negative effects. So most mutations are actually negative, but there's always that chance that a mutation can occur and be favorable to an organism, similar to what's happening in the Ames test. This mutation is actually favorable because it's changing an enzyme that no longer didn't function before, so that now it functions. And now, because it functions, the cells can grow. It happens similarly with bacteria. You have enough mutations occurring in the presence of maybe an antibiotic. Eventually, you're going to get a mutation that allows that organism to survive in that antibiotic. All right, so DNA recombination events. So this is basically when one bacteria donates DNA to another bacterium. It's kind of, in some cases, it's thought of as bacterial sex. It's kind of, I have this little piece of DNA that codes for something good, like resistance to an antibiotic, and I'm going to shoot it over to my neighbor so that he can live in that antibiotic as well. And these are done via exchanging of plasmids, or small circular pieces of DNA. And recombinant organisms, so these are organisms that contain and express genes that originated in another organism. So we can both do this in the lab, or this can happen naturally. So transmission of genetic material happens in 3 different ways, either conjugation, transformation, or transduction. Conjugation is where it's actively passed on from one bacteria to another using a pilus, so it's kind of like bacterial sex. Transformation is where the cell dies and releases its DNA. Because it's dying, it's no longer keeping its cell wall intact and all that good stuff. And the bacteria comes across that DNA and takes it in and then utilizes it. And transduction is where a virus actually incorporates that DNA into a bacterium. So you can see here conjugation is the only direct method because that's the one that actually requires the organism to facilitate the transfer of the DNA. So conjugation or bacterial sex, you have physical conjugation where you're actually creating this pilus. This pilus allows for the plasmid to be pushed into its neighbor. Also, pilus, the difference between this and this one is that a pilus is long and skinny and actually kind of like a little rod that is formed by the bacteria. Here you have just a pore, so the bacteria come into close proximity to each other, and it kind of their cell walls and membranes fuse together, opening a little bit and allowing DNA to shoot through. There's 2 different types of conjugation, so F factor and HFR transfer. F factor is where you use a plasmid, and the F positive, or the guy with the plasmid, makes a copy of it and injects it into the F negative cell, making it F positive. So you make a copy of your plasmid and inject it into your neighbor. HFR transfer is where you actually take your entire genome and copy your entire genome and then send it to your neighbor. However, the genome's usually pretty large, and this bond between the 2 cells is pretty weak, so rarely do you get the entire genome going over to its neighbor. So generally what happens is you get a little bit of the genome going over, then this bond breaks, and now that cell only has a little bit of the genome. So it's kind of like a plasmid because it's just a small little piece of DNA. And this can then be used by the bacterium that accepted it. So it's high frequency of recombination. It has an integrated F factor. So the F factor is the bit that was transferred to the bacterium. So that little bit that was transferred over actually integrates into the new bacterial genome. So chromosome genes are introduced, and or incorporation of new genes into the chromosome. So it does get incorporated via enzymes. Biomedical importance of conjugation. So we do this on a regular basis in the lab. By allowing for recombination or conjugation to occur, we can introduce DNA, naked DNA, into organisms, and those organisms can then transcribe and translate it, creating proteins that we want them to produce. Transformations. So naturally the way transformation works is bacteria die. When bacteria die, they release their DNA, and their DNA is in the environment, and bacteria that are still alive can come over, grab up that DNA, and use it. It was tested back in, I think, like the 40s, where they had bacteria that were resistant to a certain antibiotic. They then killed the bacteria by heating them, and then took that dead bacteria that was heat killed and mixed it with live bacteria that was not resistant to an antibiotic, and those live bacteria then took up the DNA from the dead guys and then became resistant to the antibiotic. Transformations we do in the lab on a regular basis using things called competent cells. Competent cells, we just mix a couple chemicals in there, a couple salts with the bacteria, and this kind of opens them up to accept more DNA. So it makes them more accepted, or they accept the DNA better. And lastly, transduction. So this is where you actually have a virus, a bacteriophage for bacteria, injecting DNA into the host. But generally so to start off the life cycle, you have a normal bacteriophage. It's injecting its nucleic acids, its viral nucleic acids, into a bacteria. Those then get preferentially chosen to be transcribed and translated into viral proteins. But for whatever reason, when those viral proteins are being produced, a few of them form viruses that, instead of incorporating the viral DNA, it's incorporating the bacterial DNA from the bacterial chromosome. So now you have 99% of the viruses coming out with viral DNA, which then can go infect another bacteria and continue on the viral life cycle. But then you have 1% of the bacteriophages that actually have DNA from the bacteria, so parts of the bacterial chromosome. And these parts can be things like resistance to antibiotics or a new virulence factor that make them more pathogenic, or whatever the case may be. So all you need is that guy that has the bacterial bits of the bacterial chromosome to go and infect another bacteria, and instead of producing virus, because it's not from the viral DNA, so the bacteria won't produce virus, instead it will take this DNA, incorporate it into its chromosome, and if it's something good, like antibiotic resistance, that bacteria is now resistant to an antibiotic. So it can be beneficial. Sometimes it can be detrimental. So it just depends on what portion of that bacteria chromosome gets incorporated into the virus and how it affects the host after infection. So specialized transduction is basically where, instead of just taking where 99% of the bacteriophages that are produced incorporate viral RNA, and then only 1% of the bacteriophages have just bacterial DNA. You have all of the bacteriophages having a little bit of viral DNA and a little bit of bacterial DNA. And this just happens by random happenstance. Generally it's because of lysogenic phases, because the viral DNA is then incorporated into the bacterial DNA early in infection. And so it makes it easier for these hybrid bacteriophages to be produced. Then these guys can then bust out, infect, or transduce their hybrid DNA, so it's both viral and bacterial, into another bacterial cell, giving this bacteria cell a little bit of bacterial DNA that may be something good, like resistance to an antibiotic or virulence factor or whatever. And then lastly, we'll talk about transposons. All I want you to know about these is they're jumping genes. So they move around. They don't stay in one location or one loci. They basically jump around to different bits and pieces, and occasionally they can jump out into a plasmid. These are how plasmids are produced. These plasmids can then be transferred to another cell. And we're done. So sorry about being a little late on this, but I've had a lot of technical issues with this. It's taken like 3 hours just to record, so don't hate me. And I will see you guys on Tuesday. Have a good